Welcome everybody to the CCMB Biologue series. Today we have the fourth talk of this series with Siddhesh Kamat from IISER Pune. CCMB Biologue is a series where our students at CCMB choose the speakers they want to listen to, and they choose experts from all across the world in life sciences. As we listen to Siddhesh today, or their work from the Kamat lab at IISER Pune, we'll see how they look into lipid signaling pathways. Theirs is one of the key labs in India that looks into lipid biochemistry. And these days they're looking into PHARC, or polyneuropathy, hearing loss, ataxia, retinitis pigmentosa, and cataract, a combination that comes through a rare genetic disorder. Something that they're looking into these days, and we'll listen to more of its details today. So, Desh, over to you.
Is my screen visible? Hello?
Yes. Yes,
okay.
I'm
audible
yes.
Too. Okay, fine. So, as the title suggests, my lab uses a chemical biology approach towards studying a human neurological disorder. Just a brief overview of our lab. There are several research themes going on in our lab, which range from looking at lipid metabolism in human diseases using animal and cell models. And we complement that with a large amount of chemical biology techniques such as organic synthesis, in some cases protein structure. And we have expertise in using mass spectrometry-based lipidomics and chemoproteomics, two techniques that I will discuss shortly, towards addressing our problems. And while we're doing that, we also couple the outputs of the information that we get from these techniques with mechanistic enzymology to understand the enzymes involved in lipid metabolism better. So if one looks at any biochemistry or any biology lab, you will find one of these posters stuck somewhere in your lab, and it basically represents an ensemble of all the pathways that are known in biology as of today. However, post-genome sequencing, it has become fairly clear that what we know in terms of cell metabolism represents very little of the cellular space, if you will. And there are a large amount of unannotated pathways that we don't know about because they might be expressed in different disease conditions or different physiological conditions. And therefore, understanding this will give us better insights into developing novel therapies for human diseases. So realizing that this is in fact a problem, the NIH in the US has over the last decade or so funded two consortiums to address this problem. One is the Enzyme Function Initiative. The other is the Lipid Maps Consortium. And both of these together, amount for more than $200 million in funding. So you can see the amount of onus that this problem is generating now. And our lab basically works in this space of unannotated pathways to understand them better to develop therapies for emerging human diseases. So as one looks at the different biomolecules, there are established genomic tools to study DNA, transcriptomics tools to study RNA, and proteomics tools to study protein. But there is a large disconnect thereafter, which ties activity to. So enzymes are a subset of proteins, and they are biological catalysts for anybody who doesn't know them. But there is a large disconnect tying in enzyme activities to their endogenous substrates and products, and there is an emerging need to develop biochemical tools to address this problem. And the two tools that our lab uses is, one of them is a chemical proteomics technique called activity-based protein profiling, or ABPP. And this uses chemical probes to address one problem. And once we identify an enzyme or a protein of interest, we use another mass spectrometry technique called metabolomics or lipidomics when it is pertaining to lipids to connect these two. So briefly, if you look at the activity-based protein profiling technique, it starts off with an activity probe. An activity probe more or less has three generic components. It has a reactive warhead, it has a linker, and a reporter tag. The warhead is something that is specific to an enzyme family, and it should label many enzymes selectively from complex biological systems such as proteomes or cells or tissues. The second property of this probe is that the labeling should be activity dependent. And the third is that the reporter tag should allow for some sort of detection, identification, or enrichment. And there are several pioneers in the field that I've listed on this slide. Ben Cravat and Matt Bojo's lab have been the first to develop chemical probes for different enzyme families. And now this is expanded to more and more enzyme families. The probe that we will be using in today's talk is a fluorophosphonate rhodamine probe. The fluorophosphonate group, which I've highlighted in red, is specific to serine nucleophiles on serine hydrolase enzymes or lipases that use a conserved serine in their active site. And the fluorophore in this case is rhodamine, but we also have a biotinylated version of this probe, which I will talk about slightly later in the talk. But I thought I will introduce an activity probe at this stage. So how does this platform work? There are two popular ways of this platform. So you have a complex proteome, you have an activity probe. You can then label specific enzymes from a complex proteome using this activity probe and resolve activities either using SDS page analysis, or you can take the same probe and append a biotin handle in the place of a fluorophore, and you can enrich proteins using Aberdeen chromatography and then analyze them by mass spec. So the gel-based ABPP platform is a slightly more high throughput platform and uses lesser time. So if you know the identity of an enzyme and you want to look at its relation in different proteomes, you would resort to the gel-based ABPP platform. If you want to identify proteins and figure out what targets for certain inhibitors or biological activities are, you would resort to the mass spectrometry-based platform. So the mass spectrometry-based platform offers you identification better than the gel-based platform. Okay, so this is about the activity-based protein profiling platform that I will describe throughout the talk. The second is, so once we have a protein of interest, we want to figure out what is its function in vivo. So we have, complex biological samples. We will subject them to different sorts of organic extractions and subject these organic extracts to different mass spectrometry platforms and establish substrate product relationships based on changes in mass signatures. So this platform, we have expanded it to about 2,500 unique metabolites encompassing these different biomolecules. Now, once we have an enzyme of interest, we will go and disrupt its activity using inhibitors or using genetic tools or using knockout mice and getting tissues or biological samples from them. And these extracts will then be subjected to different forms of mass spectrometry to get different signatures. Our lab, as introduced, focuses mostly on lipids and lipid products, and we have published extensively on our lipidomics platform. A recent paper on our lipidomics platform is shown here, where with Roop Malik's lab at TIFR Bombay, we looked at the different, lipid pathways involved in phagosomal maturation. And if anybody is interested in further exploring or reading about our lipidomics platform, here is a reference to it. I will not talk about this story today. Instead, today we will focus on PHARC. So PHARC is a human neurological disorder. PHARC is an acronym for all the symptoms associated with this neurological disorder, which are polyneuropathy, hearing loss, ataxia, retinitis pigmentosa, which is basically retinal patching, and cataract. So this is a fairly recent neurological disorder. It was only mapped in 2010 through genetic mapping efforts. And it was found that this results in mutations to this gene called ABHD12. Now, at the time this mapping was done, ABHD12 was annotated as one of 3 enzymes, along with ABHD6 and monoacylglycerol lipase, as an enzyme that can hydrolyze this endocannabinoid to arachidonyl glycerol or 2-AG to give you arachidonic acid and glycerol. This was an in vitro study and was never validated in vivo. So we will actually see what the function of ABHD12 is in vivo, and it does not really do this 2-AG hydrolysis reaction in vivo. But I would like you all to remember that this enzyme is capable of doing this hydrolysis reaction and how we can leverage it to better understand the biochemistry of this enzyme. So as I've represented on this slide, ABHD12 is an integral membrane enzyme. So it's been difficult to purify and study this enzyme, and we will show you how we assay this enzyme. All right. So little more about PHARC. PHARC is an autosomal recessive neurological disorder. There are more than 20 mutations mapped to date, all of which cause null ABHD12 expression and activity. The symptoms of this disease, appear in early teens or late childhood, and they progressively worsen with age. There is no change in the age duration that these patients live. They typically live between 65-70 years, but they are miserable throughout their life because of the progressively worsening symptoms. Now, the symptoms are early onset cataract and hearing loss with retinal pigmentation. This typically appears in patients in their mid-teenage years, and they come to a clinic with full-blown cataract. So that's one of the early diagnostic features of this disorder. Then as the patients age, they lose sense of peripheral neurons, and as a result, they have this arching of foot or loss of sensation in their hands. And eventually they end up having massive cerebellar atrophy. Here is an MRI image of one such patient, where you can see that the cerebellum has significantly shrunk. I would like you all to also remember that this disease is associated with the cerebellum, and we will see implications of this slightly later in the talk. All right. So Ben Kravat's lab made the ABHD12 knockout mouse line, where ABHD12 was genomically knocked out in these mice. And then they took the brains of these mice and subjected it to a lipidomics analysis. Now, if one did this, they found that this class of lipids, which I have drawn the structure of here, is called lyso-phosphatidylserines, or Lyso-PS as it will be abbreviated in the rest of the talk. Now, this lyso-phosphatidylserine is basically a glycerol backbone having a phosphoserine head group and a fatty acid appended to one of the glycerols at the SN1 position. Now, the numbers that you can see in the graph above on the x-axis represent fatty acids of different chains. So, for instance, if I am describing a fatty acid of 16 carbons with 0 unsaturated bonds, it will be represented as 16-0 that I'm showing with my mouse here. Whereas if I'm representing another fatty acid with 22 carbons and 4 double bonds or 4 unsaturations in it, it will be represented as something like 22-4. So this is a common nomenclature for all fatty acids, and this is a nomenclature that we will be using for the rest of the talk. Okay. So what was found was that in the brains of ABHD12 knockout mice, levels of lyso-phosphatidylserines significantly increased. Okay, in particular, these very long chain ones, which are carbon 20 and higher, was significantly elevated. Okay, so the mice also showed that an age-dependent neuroinflammation, which was an increased microgliosis in the cerebellum of these mice. And further, these mice failed different neurobehavioral tests that are routinely done to check neurological function. Okay, so in principle, these mice kind of exhibited PHARC-like symptoms of human patients. All right. Another take-home message from this slide is that these very long-chain lipids significantly are elevated, and we will get back to this point based on this introduction slide. Okay. So now, what was the hypothesis for PHARC? The hypothesis was that the lyso-phosphatidylserine lipids significantly elevate following ABHD12 deletion. This results in microglial activation and thereby neuroinflammation, and that eventually manifests into neurobehavioral dysfunction as a result of increasing age. Now, the interesting thing was whether there is any PS lipase that can make these lyso-PS lipids, and we will also look at this slightly later in the talk. Okay. Now coming to what our lab has done with this problem. So we've seen that ABHD12 is a lysophosphatidylserine lipase. Okay, it does a lipase reaction where it cleaves off this fatty acid at the SN1 position of a lysophosphatidylserine group to give you free fatty acid and the glycerophosphorine head group. What I also mentioned from the introduction slide is that ABHD12 can also do monoacyl glycerol lipase activity or MAG lipase activity. It can hydrolyze both two MAG substrates like we saw with 2AG, and it can also hydrolyze one MAG substrates like reported in different systems in literature. Okay. Now, both these lipids you can buy commercially, but they come with limited fatty acids appended to it, mostly palmitic acid and stearic acid, which is C16 and C18. But if you want to screen a library of these lipids, they are not commercially available. So what we decided to do was to synthesize the library of 1-MAG lipids. And being from a biology department, we like to keep our chemistry synthesis very, very easy and get very simple tractable chemical compounds. So what we did was we started off with this commercial protected glycerol group, which you can buy, and you can esterify it with EDC coupling to give you this intermediate. And you can easily open up this intermediate using acid hydrolysis to give you 1-MAG lipid libraries. So we basically played around with this lipid part of the molecule, and we made a library spanning lipids from carbon from a C10 chain all the way to a C24 chain. Okay, so while the chemists were making this lipid library, the biologists in lab were exploring the effect of glycosylation on ABHD12 activity. So ABHD12 is a glycosylated enzyme, and before we figured out what the substrate preference for this enzyme was, we wanted to see whether glycosylation of this enzyme is, if at all, required. So as you can see in this first set of data that I'm showing, this is a typical activity-based gel profile where we have a wild-type enzyme, we have a catalytically dead serine to alanine mutant. And you can see they express at nearly equal levels in overexpressed mammalian cells. And if you take the membrane lysates and you assay them for activity using an ABPP gel-based ABPP experiment, you will find that the wild type labels very well with an activity-based probe, but the serine to alanine mutant is virtually dead. Okay. Another thing we did is we took the wild type enzyme, we pre-labeled it, and then we deglycosylated it using PNGSF, and you can see a nice shift in band showing us that our overexpressed enzyme is indeed glycosylated, and we can show this off the gel as well. So if you now take this glycosylate, if you now check the effect of glycosylation on ABHD12 activity, what we find basically is that if you remove off the glycosylation, the enzyme activity significantly gets depleted. So what that means is that ABHD12 requires glycosylation for optimal lipase activity. Fine. So now we decided, therefore, to do all our assays with the glycosylated form of ABHD12 that we are expressing in mammalian cells. So we then checked whether MAG substrates were comparable to Lyso-PS, and while the the Vmax of these different substrates are slightly different, with Lyso-PS being the best, the KMs were comparable. So that told us that we can, in principle, use MAG to inform us on the enzyme selectivity across different fatty acid chains. And we did that enzyme kinetics assay, and we found that ABHD12 prefers very long chain lipids as substrates, okay, which kind of ties in very well with the very long chain Lyso-PS that accumulate in the brains of ABHD12 knockout mice. So this kind of provides a biochemical explanation as to why these lipids are accumulating in the brain. It is because this enzyme prefers this class of lipids over long chain or medium chain lipids. All right. Now we also decided to test. So this experiment was done with an overexpressed membrane lysate that was expressing ABHD12 in the wild type form. But we also wanted to assay what endogenous mouse brain ABHD12 prefers. Now there was a slight complication here because the MAG lipase activity from the brain comes as an ensemble of an activity of 3 enzymes: ABHD6, monoacyl glycerol lipase, and ABHD12. Thankfully for us, Ben Kravat's lab has made very, very good probes or very, very good inhibitors for ABHD6 and monoacyl glycerol lipase. So we could use this in our system to specifically look at the contribution of the monoacyl glycerol lipase activity coming from ABHD12. So we first confirmed that ABHD12 in the mouse brain was in fact active. We show here using knockout brain lysates that yes, ABHD12 is active by a gel-based ABPP experiment, and it is gone at a protein level in the knockout mouse brains. And when we again assay our entire library against mouse brain lysates from wild type or ABHD12 knockout brains treated with ABHD6 or monoacyl glycerol inhibitors, we find that the endogenous ABHD12 again prefers this entire very long chain lipid substrates. Interestingly, we find that unsaturations in the very long chains do not affect the activity, and the activity for this enzyme towards very long chain lipids is exclusively governed by the chain length. All right. So why is this finding interesting at all? So to understand this a little more physiologically, what Alomi and Shubham from lab did is they took different tissues or cells and they fractionated it into different subcellular fractions. And what they found was ABHD12 was significantly enriched in this microsomal fraction. And if you do immunofluorescence experiments, we found that ABHD12 was mostly localized to the endoplasmic reticulum membrane. Now, why is this finding of any relevance? Is because if you look at synthesis of very long chain lipids in the cells, you find that in the cytoplasm, there is an enzyme called fatty acid synthase that can, in principle, only make versions up to carbon 16, which is palmitic acid. And anything beyond that is made by a set of enzymes called elongases, which are localized in the ER membrane. So what we think is happening is while all these very long chain fatty acids are being made, ABHD12 is controlling its secretion from the liver by performing monoacyl glycerol lipase activity or Lyso-PS lipase activity. And its absence is going to result in a significant increase in secretion of these very long chain lipids in these various forms. So a part of this finding is now published in the Journal of Biological Chemistry. Here is the reference to the paper about the findings that I have described so far. This paper made it to the cover of JBC. And if anybody is interested in reading it further, here is the reference. Okay. Now, as I said, ABHD12 is a Lyso-PS lipase. Okay, so it basically hydrolyzes lysophosphatidylserine lipids to give you free fatty acid, and its absence causes the human neurological disease PHARC. Now, we wanted to also understand whether an upstream enzyme, which is ABHD16A, that converts PS to Lyso-PS or phosphatidylserine to lysophosphatidylserine by hydrolysis of this fatty acid, has any role in the amount of lysophosphatidylserines in different brain compartments. Okay, so what Shubham did first is he looked at the localization of ABHD16A, and he found that this enzyme is also present in the ER. And interestingly, if you look at the membrane orientations of the active sites of both these enzymes, you find that ABHD16A faces cytosolically, where all the phosphatidylserine is enriched, while ABHD12 faces luminally, where we think a lot of Lyso-PS signaling happens and Lyso-PS secretion happens. So he showed that there is an interplay between these two enzymes and found a new pathway for PS metabolism at the ER. Okay. Now, following up on this finding, we, not being a neurobiology lab, used a lot of help from Vidita Vaidya's lab at TIFR Mumbai and Professor Subedar, who is a faculty at IISER Pune. And they showed Shubham how to get different anatomical components of the brain. So Shubham was able to get different anatomical components of the brain. And he found that this enzyme ABHD16A is significantly enriched in the cerebellum, whereas ABHD12 is more or less ubiquitous across different brain regions. All right. So he corroborated this protein expression levels with substrate hydrolysis assays, and he found that the PS lipase activity was highest in the cerebellum as compared to any other brain component. Okay. He also confirmed this using different immunofluorescence experiments, which I will not show you. What he also did was consistent with ABHD16A, which is the major biosynthetic enzyme for making Lyso-PS lipids. He found that consistent with its increased expression in the cerebellum, the levels of lysophosphatidylserine lipids were also significantly up in the cerebellum. Okay, so this kind of is a first indication as to why the cerebellum might be the most affected part in PHARC, okay, in the brain. Now, following up on this finding, what Shubham did was he again looked at different Lyso-PS lipids in the different brain anatomical regions, and he looked at the effect of ABHD16A deletion and ABHD12 deletion on the levels of these lipids. So he found that if you delete ABHD16A, if you focus on lane 6, you will see that lysophosphatidylserine levels significantly go down, and phosphatidylserine, the substrate levels go up. Kind of an indication that this correlates very well with the expression and activity of ABHD16A in the cerebellum. Conversely, if you look at ABHD12, which is more ubiquitously expressed, you find that everything goes haywire if you delete ABHD12 in the brain. The effects of ABHD12 are again most profound in the cerebellum, consistent with its upstream enzyme being most localized there. So, with all this information, we decided to further look into the cerebellum to figure out whether anything was unique here. And what Shubham found was that if you look at the different regions of the cerebellum, again, we are no experts of this, and we use a lot of advice from a lot of neurobiologists, and Shubham talks to a lot of them. What we found was that ABHD16A is mostly expressed in the granular layer cells of the cerebellum, and to a certain degree, even in the molecular layer cells. Conversely, if you look at ABHD12 now, ABHD12 is exclusively expressed in the Purkinje neuron layer of the cerebellum, and it's more or less absent in the other two layers. We also confirmed this using the respective knockout mice. So I'm not showing you all the information, but the bottom line was that ABHD16A was present in granular layer cells and ABHD12 was present in the Purkinje neuron. And now, based on this finding, Shubham came up with a hypothesis that the granular layer cells of the cerebellum, where ABHD16A is situated, are constantly making these Lyso-PS lipids. And for homeostatic signaling, ABHD12 is regulating their levels in the brain through the Purkinje neurons. And if ABHD12 is deleted in these Purkinje neurons, you see a buildup of Lyso-PS lipids, and thereby you see increased signaling coming from this class of lipids, and therefore you're possibly seeing all these neurological symptoms in this disease. So we kind of think that there is a paracrine signaling model towards Lyso-PS signaling in the mammalian brain, and it is more or less coming out of a focused center in the cerebellum and possibly spreading to remaining parts of the brain. Okay, so this is more or less a fresh of the mill paper that is up in biochemistry a month or two ago. And here is the reference to the findings from this paper in case anybody is interested in reading. Okay, now I'll switch gears a little and I'll come back to PHARC at the end of the talk again. Okay, so this is kind of our understanding biochemically of PHARC on a project going on in lab. Independently, my lab is also interested in looking at oxidative stress in mammalian cells. Okay, so oxidative stress, as most of you know, is an imbalance between I can't hear. Hello?
The slides are not moving. We are still stuck at cerebellar localization.
Can you see this?
It's moving fine.
No,
Probably
it is fine.
It's something to
it's
do
going,
with your internet connection.
Yeah, it's moving fine. Yeah,
Okay.
it's moving fine. Yeah, for others.
Yeah. Okay.
Okay, so a part of lab is also looking at oxidative stress in mammalian cells. And as most of you all know, oxidative stress is an imbalance between the cellular redox components in the favor of the oxidants. And oxidants are mostly these reactive oxygen species or free radicals, and they're known to damage different biological molecules. There's lots of study about oxidative stress in the context of DNA damage or nucleic acid damage and protein damage. But you'll be surprised that there is very little known about its association with lipid oxidation and what that means in terms of physiology. So this was something that we decided to address upfront. And it's been challenging to look at this in the context of lipids, because if you take a lipid, this is a single fatty acid with one double bond. And if you add a reactive oxygen species such as superoxide or hydroxy radicals, you will find a whole bunch of products form, and you have no control on how these products are forming in a reactive oxygen species based condition. So that makes it even more challenging to figure out which of these are physiologically relevant and what they're doing. The other complexity is, remember now, these lipids are also associated with different fatty acid head groups, and there is a crosstalk between these head groups. So that further makes it difficult to identify what are all these species in a physiological setting. So what we decided to do was to simplify this system a little, and we decided to look at only phosphatidylserine lipids. So why did we choose this is because if you look at a normal cellular membrane, which is the bilayer, all the phosphatidylserine lipids are facing cytosolically under normal conditions. Okay, and you will not find any phosphatidylserine on the extracellular surface. However, when cells is subjected to reactive oxygen species or oxidative stress, this phosphatidylserine membrane asymmetry is broken, and a lot of PS gets externalized. Okay, the reason this happens is because the unsaturated fatty acids esterified to phosphatidylserine head groups get oxygenated or oxidized, and therefore they cannot be accommodated in the normal membrane curvature, and therefore they flip. A lot of immunologists use this as a means to look at cells undergoing apoptosis, because it is this externalized phosphatidylserine that is recognized by the annexin 5 antibody. Okay, while we know all of this, the actual chemical structure of these phosphatidylserine lipids which are flipped across the membrane bilayer remain unknown, and this is something we wanted to figure out. Okay, so this is also initiation of your programmed cell death or apoptosis, as I said, used by a marker by immunologists to assess this. So we decided to approach this using a two-pronged approach. We wanted to see what is happening in cells or tissues, and we also wanted to figure out whether this matches something we can make in a test tube. So to figure out whether we can generate oxidative stress in cells in a controlled manner, we collaborated with Harinath Chakrapani's lab, who is a faculty in chemistry at ISER Pune, and his student Ravi. And what Ravi made is this compound MGR1. And what MGR1 does is, once it gets into cells, it is acted upon by cellular esterases to make this molecule JCHD, which reacts again through the cellular redox machinery to give you superoxide in the end. And what we also decided to make is a structurally similar compound without the juglone scaffold, and this would serve as a good control for any off-target effects or toxicity effects that might come from MGR1. So what we found was that as a function of esterases in a test tube, MGR1 robustly produced superoxide, whereas MGR2 did not. And when we treated cells with MGR1 or MGR2, we found that MGR1 significantly produced reactive oxygen species in cells, whereas MGR2 did not. So just to give you a comparison, we found that approximately 25 micromolar of MGR1 treatment produced the same amount of ROS as 1 millimolar treatment with hydrogen peroxide. So it was fairly robust because this molecule can undergo multiple turnovers to give you these reactive oxygen species. So you don't need too much of it, and you have more control about how much you're adding in the system. So while the chemists and biologists were working on that, two folks from my lab, Amol and Nilay, were trying to figure out what happens when you take phosphatidylserine in a test tube and abuse it with reactive oxygen species. So the phosphatidylserine that we chose was one that had a C18 fatty acid, which is a saturated fatty acid here, and a C18-1 fatty acid, which is oleic acid, which I'm showing here. Okay, so we anticipated that this double bond of oleic acid would get oxygenated in some way when you add different reactive oxygen species to it. So what we found was that we found more or less two stable versions appear at the end of this experiment. We found a version that we called Ox 18018-1 PS. It basically had a plus 16 mass addition coming from an epoxide or an oxygen added across that double bond. And parallelly, we think what happens is that this epoxide gets hydrolyzed in some way, and you end up with this HY version or a hydroxy version where it turns out that you have a plus 18 mass addition, similar to addition of water across that double bond. Okay, so now we found that from a starting PS lipid with having a unsaturated bond in the lipid tail, we could generate two stable versions of it in a test tube. Now, do these versions actually exist in cells was the question. And we found that when cells were treated with MGR1, you found a significant accumulation of both these species in different cells, whereas MGR2 or DMSO produced very, very little amounts of this. Interestingly, based on this, we generated a mass spectrometry method where we hypothesized that the SN2 chain, which is this chain, if it has an unsaturated fatty acid or more than one unsaturated fatty acid versions, we hypothesized that we would end up having these epoxy or this hydrogenated versions of these oxidized phosphatidylserines. And indeed, that is what we found, is that when we treated cells with MGR1, which are the bars shown in red, you will see a significant accumulation of all these oxidized phosphatidylserine lipids, whereas these are very, very low and at basal levels in MGR2 treated cells. So now we had a very good way of generating reactive oxygen species in cells in a controlled manner. And we had a good mass spectrometry method to detect these oxidized phosphatidylserines in different mammalian cells. So what we decided to do was to develop a chemical genetic screen. So what we did is we collated a library of known lipase inhibitors, which can be commercially bought or they were given to us by Ben Cravat and other labs. And the idea was that if you take cells and you treat them with these known lipase inhibitors, following which you generate ROS in these cells and you measure oxidized phosphatidylserine levels, if these inhibitors are indeed inhibiting lipases that hydrolyze oxidized phosphatidylserine, their levels will go up in cells. Okay, so we performed this entire screen and we found that most of the compounds were completely inactive in this screen. We, however, found that three compounds, MAFP, THL and FP alkyne produce more than twofold increase in oxidized phosphatidylserines in different mammalian cells. So if you look at MAFP and FP alkyne, both of them have a fluorophosphonate head group. So what this told us was that the likely enzyme is belonging to the serine hydrolase class of lipases, and it is most likely a membrane-bound enzyme based on the localization of these oxidized PS lipids. Because MAFP and FP alkyne together have numerous targets, we decided to focus on THL, okay, as a tool to figure out what this enzyme is. So THL is tetrahydrolipostatin. It is a general lipase inhibitor, and in the US it is sold as an over-the-counter drug called Orlistat to treat obesity, more or less. What it does is, if you ingest it orally, it inhibits a bunch of pancreatic lipases and prevents absorption of triglycerides. But it is also used across different cellular systems to identify different lipases. And THL has only a handful of targets. So we went in, and what we did was a competitive chemoproteomics experiment where we took proteomes, or in this case, we took cells. We treated them with THL. THL will form a covalent adduct across the serine, so it will no longer be reactive to the activity-based probe because THL is going to block the active site. And then in a competition experiment, we would figure out what the targets of THL are. So if one looks at this profile using a gel-based platform in complex proteomes, we found that in soluble lysates, as expected, there were no real targets of THL. However, in the membrane profile, we found several targets that are losing activity as a function of THL treatment. I'm sorry that this gel is a little blurred. I was hoping to have a slightly better gel, but I don't have one. But we also did the same thing using mass spectrometry. And what we found was that THL had about 10 targets or so where the inhibition was greater than 90%. Okay. We then pulled out all the lipases that THL was interacting with. We overexpressed all of them and measured oxidized PS lipase activity for all these lipases. And interestingly, we found that ABHD12 that I have described earlier in the talk had the most robust oxidized PS lipase activity, whereas ABHD16A, which is a PS lipase, also a target of THL, has no such activity. Okay, so this was an interesting finding for us that ABHD12 is likely an enzyme that's going to control oxidized PS lipase activity. So if you compare the the oxidized PS lipase activity of ABHD12 along with the lyso-PS lipase activity of ABHD12, we find that they are comparable, and this enzyme has no activity against phosphatidylserine. Now, if we take cells that are overexpressing ABHD12, the wild type form, and we subject these cells to oxidative stress, we find that ABHD12 overexpression results in significantly decreased oxidized phosphatidylserine lipids, whereas a mutant does not do that. It has almost mock levels of activity. So ABHD12 activity, increase in ABHD12 activity, in fact, reduces oxidized PS lipids in cells. We can also do the converse, where if you block ABHD12 activity or delete ABHD12, you find an increase in the levels of these lipids in cells. Now, at this point, we submitted a paper and the reviewers asked us for something that was more physiological, which was a correct criticism, because nobody has really seen these lipids in different organs or tissues. So what we did is we took wild type or ABHD12 knockout mice, and we found that a systemic lipopolysaccharide injection increases neuroinflammation or increases oxidative stress in the brain. And what we wanted to do at the end of this is take out the brain and see whether we can see oxidized PS lipids. What we found was that if you take wild type mice, basically under normal conditions, you don't see these lipids. Whereas if you stress these mice with LPS, you do see an increase or a slight accumulation of these lipids in the brain. Now, if you do the same experiment in ABHD12 knockout mice, you find that the levels of these lipids are significantly higher compared to a wild type mouse. Okay, so this kind of provides a in vivo physiological understanding that oxidized PS lipids increase in the brains of mice that have reduced ABHD12 activity. Fine. Now, this is another thing that we found really interesting from the study is if you take these lipids and you just sprinkle them on primary or cultured macrophages, we find a significantly increased response for these macrophages towards secreting pro-inflammatory cytokines. So if you compare ABHD12 knockout samples, they almost have LPS levels of stimulation in terms of secretion of these pro-inflammatory cytokines. This is a shout out to any immunologist out there who's interested in helping us figure out what the receptors or how this signaling happens, because we don't know how this happens. So coming back to the question is, why does ABHD12 perform this oxidized PS lipase activity and not ABHD16A, which is a known PS lipase? So we think it is because of the membrane orientations of these two enzymes. ABHD16A faces cytosolically, whereas ABHD12 faces extracellularly or luminally. And most of the PS under normal conditions is present on the cytosolic face of the membrane. Now, when you subject a cell to a reactive oxygen species, you find that these PS lipids get oxygenated and they flip their membrane orientation. And ABHD16A can no longer see these lipids, and because the active site of ABHD12 is there to see these lipids in a physiological setting, we think that is why ABHD12 is an oxidized PS lipase. So how does this add to our understanding of PHARC? Now, coming back in time this to PHARC, what we think is that under normal conditions, the PS lipase ABHD16A is converting PS to Lyso-PS, and increase in Lyso-PS following ABHD12 deletion is resulting in some pathology of PHARC. Now, what we found across reading literature is that a lot of oxidative stress is involved in a lot of neurological conditions. And we think along with the Lyso-PS lipids, oxidized phosphatidylserine lipids are also being transiently produced, not at a homeostatic level, but probably in bursts as patients undergo stress. And that, along with Lyso-PS lipids, is likely contributing to the pathology of PHARC. Okay. The second part of the talk on the oxidized phosphatidylserine lipids was published last year. Here is a reference to it. It came out in Nature Chemical Biology, in case anybody wants to go back and read the paper and more findings on this topic. So where does that leave us now, and where are we headed? So we've seen that ABHD12 controls levels of lysophosphatidylserines and oxidized phosphatidylserines, mostly in the brain and in primary macrophages. But we find that both these activities and these lipids are present in different tissues. So we think there are other lipases beyond ABHD12 that are controlling levels of these lipids in different tissues. And we have some preliminary evidence that these enzymes are belonging to the serine hydrolase family of lipases. We don't know what the exact identities are yet, but it's good to know that approximately 75% of this family still remains unannotated. And it gives us a lot of opportunity to explore what these members might be. Okay. Now, if you look at these lipids generally, I haven't spoken about their immunological outputs, but these lipids are known to regulate several facets in immunology. The most studied is its role in mast cell degranulation, where it stimulates histamine release and therefore an anti-allergic response. It is also responsible for macrophage activation, and it is also responsible for maturation of regulatory T cells. Okay, but there has also one more thing that is kind of known putatively, though not fully validated, is that there are 3 potential receptors for the Lyso-PS class of lipids. They belong to the P2Y family of GPCRs. And there is one paper where they express pretty much all GPCRs and they're trying to figure out what the ligands are. And these three receptors came out as being receptors to Lyso-PS in an in vitro screening model, though in vivo studies are still remaining to validate this. In particular, GPR34 has been shown to some extent to facilitate mast cell degranulation, but the role of others are still speculative at this point. Now, studying Lyso-PS immunobiology becomes challenging at times because of several reasons. One is that these lipids are commercially very, very sparse. You get it from maybe one or two vendors, and most times they're back ordered. They're exorbitantly expensive to buy and do studies on this scale. Secondly, most of the receptors that I spoke about before, it's hard to find these receptors in cultured cells. So they exist in primary cells. You can get them from PBMCs or you can get them from macrophages, but surrogate lines that you routinely culture seem to lose these receptors over time. The third is that of the three receptors that I spoke about, macrophages have none of them across different types of macrophages. So what are the receptors in macrophages is also unknown. So what we are trying to do is we are trying to first synthesize this class of lipids, lysophosphatidylserines. It's been challenging to synthesize this because, as you can see, this particular lipid has a sugar, an amino acid, and a lipid tail. So you can see it has a combination of hydrophobic and hydrophilic groups, and therefore it's difficult to build them together. What we plan to do once we eventually make these lipids and we are very close is we want to play around a little bit with the fatty acid tail group and add different appendages to it, such as a photo cross-linking group or a group that will allow us to enrich it like an alkyne group that will enable click chemistry to pull down things that we can photo cross-link this lipid to and figure out what receptors or protein ligands are for Lyso-PS in different mammalian cells. Okay, so these are a few thoughts that I'm leaving you all with. At the end, I would like to acknowledge several people who have contributed to the study. All the members of the current lab have contributed to the things I've spoken about in some way or the other. Certain people who have recently left us for better positions are also involved in these studies. I would especially like to thank several collaborators, Hari, whose lab we collaborate with to do a lot of chemistry, Vidita, whose lab we go to for all neurobiology related things, along with Professor Subedar. I'm also thankful to Roop, who's recently moved to IIT Bombay. His lab kind of is a prototype for testing all of our lipidomics platform. And Satyajit and Vinita from IISER Pune, who guide us a lot on the immunology part and are very patient with us asking very stupid questions to them all the time. We are also fairly active on Twitter in case anybody wants to follow our research. I would also like to thank all the funding agencies for giving us the opportunity to do these studies. A big thank you to the organizers of the CCMB Biologue for organizing such a wonderful event. I've also listed my email here in case anybody has any issues connecting. You can send me an email regarding any of our studies described today. So that's all I have. I'm open to any questions.
Yeah, Siddhesh, Shankar here. Can I begin?
Yes, go ahead.
Asking some of the stupid questions that you may have asked.
Sure.
So I'll begin maybe with one of them. So the point that you are trying to say is there are 3 enzymes at least doing this job, correct? In terms of the basic enzymatic activity.
Correct.
And you say that because of the lipid profile, correct, that these enzymes exhibit, that
Yes.
ABHD12 is uniquely poised to do this. Or do you have such promiscuity in the other 2 enzymes as well?
So
that's something which did not, I couldn't, yeah.
see if I can exit and go back to that slide.
Because you were quite clear in terms of the ABHD12's lipid profile, you know, in terms of its activity, but
Okay.
I was not sure about the other two.
These three enzymes, okay, what I forgot to mention is they have different orientations in terms of the active site. Okay,
Sure.
so monoacylglycerol lipase is a soluble enzyme floating around in the cytoplasm. ABHD6 is an integral membrane enzyme with a cytosolically facing active site, and ABHD12 is the only one that is extracellularly facing. Okay,
Sure.
so all of them can do the monoacylglycerol lipase activity. However, ABHD12 is the only one that does the lysophosphatidylserine lipase activity.
Okay, the others do not have that.
So there is promiscuity on the monoacylglycerol lipase activity, but not the other one.
The other question that I had linking to this ABHD12's enzymatic profile is that you said for different chain lengths you have only a Km effect, correct? But that was a little funny because you have different acyl chains which you would expect to have some K, you know.
So I'm comparing the Kms only for this fatty acid group.
Okay.
So for a constant fatty acid group, if you vary the group, the Kms are comparable. Okay. But if you look at the fatty acid chain lengths, you would find that the Kms decrease for the very long chain fatty acid.
Okay.
So there are two comparisons here. One I'm comparing against.
Sure. Okay. I got it. Yeah. So that's clear. Yeah, because that was the confusion between the first and the second one in terms of discriminating. Okay. Thank you, Siddhesh. Yeah, it was a great talk.
Thank you.
Yes, Chandak.
Yeah. Okay. So Siddhesh, yeah, very nice talk. I
can't hear you clearly. You're breaking up.
Chandak, we can't hear you, Chandak. Maybe you should type the question, possibly. You can put your question in the chat box and maybe somebody can read it. We can't hear it.
Yeah. Okay.
Yeah, now no one is asking, maybe I can ask another question. This flipping of lipids based on oxidative stress that you mentioned, it can happen on both sides, correct?
Can you repeat, Shankar?
Yeah, sorry. So the flipping of these lipids, you showed that it happens only on one side where this, ABHD12, performs its activity. But can it also happen on the inside, which is the cytosolic side, and how that is handled?
So we don't know whether it happens on the inner leaflet, because there are a lot of these scramlases and a lot of these flipases that tend to push anything oxidized into the extracellular space.
Okay, so that is unidirectional. I mean, basically on one side only.
It seems to be unidirectional, but nobody knows whether it actually flips inside the cell also. It may flip and it might be so fast. So there are promiscuous esterases that are floating on in the cytosolic soup. So the hypothesis is that those may be just hydrolyzed with the promiscuous esterases.
Okay. And this ABH12, it's actually expressed majorly in neuronal cells, is it?
It's mostly present in the nervous system. It's present throughout the brain, as we showed, and it's also present in different macrophage cell lines.
Okay. I see somebody else is raising their hand. Yeah.
Yeah.
Am I audible now?
Yes, Chantal. Your question is
okay.
also here. If you want, I can just read it out if that makes it easier.
If I'm audible, I'll just say, I mean, Siddhesh, nice talk. And you said that there are 20 mutations that are identified in PHARC. And so I was wondering whether all of these are null mutations or any of these actually lead to reduced enzyme activity. Because most of the experiments you have used is like, knockout, and they show that ABHD12 activity is completely lost. So is there any study which has looked at a reduced enzyme activity because of specific mutation and correlated with any phenotypic heterogeneity?
So two answers. One is that all the mutations mapped in humans result in null ABHD12. So you do never get the full length protein. Now, if you look at the architecture of this protein has, I think, one or two N-terminal membranes that lodge it to the different cellular membranes, and the actual catalytic active site is very far towards the C-terminus. So most of the mutations generally happen very early on in the N-terminus, and they result in a stop codon very early on. So you never get the full length protein. Okay, in humans. So coming to your second question, if you, let me just go back to one of my introduction slides. So if you look at reduced ABHD12 activity, so we have also looked at heterozygous animals, which have 50% of ABHD12. And they, for all practical purposes, behave like wild type animals in terms of lipid levels, in terms of behavior, in terms of everything. So you need complete knockout of ABHD12 to start seeing effects.
Okay. And are there any ABHD16 mutations known? And if yes, then what is their phenotype?
So ABHD16A I haven't gone into, but ABHD16A in humans, to the best of my knowledge, there are no mutations reported. There can be no mutations reported because ABHD16A mice, ABHD16A knockout mice have a high degree of embryonic lethality.
Okay.
So I don't know whether people with ABHD16A mutations are alive.
Okay. Just one last thing, that is there any such case reported in India, or is there any idea that what's the prevalence of this disease in this country?
So PHARC by itself, you will not know that it is PHARC till you actually do genetic mapping. Because if you look at the symptoms, they are fairly garden variety neurological symptoms. So figuring out you have PHARC, you actually have to do genetic, genetic typing. And I know of one case that has recently come across because the PHARC people kind of are trying to build a consortium because they think it is more prevalent than it is thought to be. So they just want to know these things. We will because it's only known for 10 years. It is still fairly infant. We are trying to develop some sort of blood biomarkers with all these groups to figure out whether we can do something about it.
Yeah, thanks. I'll be in touch with you. I have some other questions.
Sounds good.
Yeah, thanks.
Can I ask? This is Santosh.
Go ahead.
Hello.
Yes.
Yeah. So I have two questions. First one is regarding the oxidative stress. So even in a normal cell, there is a basal oxidative stress. But when you look at your oxidative stress levels, nothing happens to the lipids. And if you compare with proteins and nucleic acids, where do lipids stand?
So let me address your first question first. So if we come to this slide, so if you look at basally, we do see a little bit of oxidation. So if you look at a DMSO control, we do see a little bit of lipids form, but because we are using very high-end mass spectrometry and very sensitive instruments, we can see it. But there is basal lipid oxidation, and it's probably at a homeostatic level where the cell can deal with it. Okay. Now, if you look at, your second question was where do lipids stand in terms of proteins and nucleic acids. So if you look at it as a game of chess, Lipids would be your pawns. They get killed first. You then start seeing damage to proteins, which are your other players, and then probably your DNA and things in your nucleus are your king, which once it gets damaged, you're dead.
Oh, and did you look at the oxidation with respect to aging? Because
We
with
have
respect
not.
to
We
aging
have
and all, we know that.
No, we have not. That's something in the works.
Yeah. Thank you.
Can I ask one more question, Siddhesh?
Yes, go ahead.
Yeah. So you showed that ABHD12 activity is, maximum in the cerebellum. So most of these accumulations and phenotype like ataxia are related to cerebellar atrophy. But if we look at the expression, it is also there in the olfactory bulb and various other regions. So, but the phenotype which comes is not exactly commensurate. So, what explanation you have for that?
So ABHD12 is more or less ubiquitously expressed. Okay, so ABHD12 is the enzyme that's going to degrade this lipid. The enzyme that's going to biosynthesize this lipid, which is ABHD16A, is more prevalent in the cerebellum and to a lesser extent in the olfactory bulb. So the effects that you're seeing are mostly because of the biosynthetic enzyme making this lipid on a higher flux in the cerebellum. So the accumulation and other things are going to start off in the cerebellum and then percolate elsewhere. So that's why we think that the cerebellum is the region that's probably damaged first and most.
Yeah, I mean phenotypes like retinal pigmentation and others, they are very specific to the region. So I was just wondering, and we don't have much of smell loss or anything, although olfactory bulb is the second major one where the ABHD16A is expressed.
The second phenotypes are again all in the eyes. There is cataract. So again ABHD16A is expressed there. So I don't know whether there is a correlation. I am at best a hand-waving neurobiologist, so I wouldn't be able to give more insights on this.
Yeah. Thanks a lot.
Hello, Siddhesh sir.
Yes.
Yeah, very nice talk. So my question is pertaining to the activity of ADH12. I mean, there are, three activities which you have mentioned in the talk, like MAG lipase, Lyso-PS lipase, and Oxidized PS. It can hydrolyze. So and then the mutants of ABH12. They also have association with PHARC. I mean, all these symptoms. So, is there? Do you have any insights about which of the three activity? Is related to the PHARC, and is it like combination of? All these three, or some one activity has higher or lower? This relation to these phenotypes.
Okay, so of the three activities. If you look at the levels of these lipids, first of all, there is no change in monoacylglycerol lipids in the brain in any context. So it is unclear whether physiologically ABHD12 is doing a monoacylglycerol activity. It may be doing this in very confined pockets or in specific contexts. But generally, there is no evidence pointing to ABHD12 being a major monoacylglycerol lipase. Okay, so while it does the activity in vitro, there is no evidence of it doing it in vivo. All right. The most profound thing that is always found is that ABHD12 deletion always causes accumulation of Lyso-PS lipids. So I'm showing it on this current slide, where Lyso-PS lipids are always accumulating. So ABHD12 is definitely a Lyso-PS lipase in vivo. Now, under cases of oxidative stress, so patients have some stress, or they have some injury or something like that. Under those conditions, when levels of reactive oxygen species go up, you have these oxidized PS lipids also build up in an ABHD12 knockout system. So we think it is a combination of lyso-PS and oxidized PS that is probably contributing to the pathology of PHARC.
Okay, sir. Yeah. Yes. Thank you. Yes.
Siddhesh,
Yes.
I wanted to know how far back in on the evolutionary scale does lipase go? I mean, ABHD12 to be specific, you can probably answer. Or I'm basically coming from a space where I want to understand how far back can we date the lipid protein signaling merging with each other.
So lyso-PS lipids are more or less, described